Right, so the website is intended to be very simple to use. You can just type in a single gene, like Emma did. But you can also go a little bit beyond that, and let's say you have a set of genes. So in this case, I was involved in a study of Alzheimer's disease, and that study gave me 4 genes that were modified in this study. So you can paste multiple genes here in the query box. And then what you might want to ask first is, are these genes then connected to each other in the network? So then you can expand this advanced search menu here. And there's quite a lot you can do there actually. So I'm not going to show everything here, but there's one. So you can change the threshold of confidence, of course. If you want to sort of look for less confident things, if you don't find anything in the first search, you can lower that a bit. The expansion depth is quite interesting. So normally it's set to 1, which means find the nearest neighbor. So and that will sort of give you the extended network then. But if you set it to 0, it won't look for other genes than you have in the query set. So by doing that and then pressing search, it will just look for through the network for the genes in the query. So that's so in this case, it's quite nice. They were all connected to each other in this particular way. Sometimes you can do this for maybe if you have you can also do this for like 50 or 100 genes or more. Then you might get specific clusters when you do this. So it can be quite informative. Okay, but so I'm not going to show more of that. Instead, the idea is to show this thing that Dimitri mentioned in the introduction on comparative interactomics. So this is something you cannot do, as far as we know, on any other website. And that is, you can take your query and then you can ask FunCoup to look for conserved subnetworks of orthologous genes in other species. So since this is Alzheimer's, C. elegans might be a good model organism to pick here if we want to sort of do experiments later on. And then so you just do that and press search. And since I already did that, it takes 10 seconds. You get this mixed network then of the 2 species. So human in blue, C. elegans in red. And and then you get 2 types of links also. So you get connections between the species as dashed green lines here, which are so these are orthologs then in the paranoid database, like this. And yeah, and then you can look at this in many different ways. In particular, so these nodes here with bold outlines are the query genes. So these are my 4 query genes up here. And then presenilin 1 has an ortholog called cell12 in C. elegans. So what you can look for then is, are there any interactions of PSAN1 that have orthologs that are also interacting with cell12? Well, this is an example, for instance. So TMED2 is orthologous to cell9, which is then interacting with cell12. So this forms sort of a rectangle of 2 interaction links and 2 orthologous links. So that's a very strong suggestion that whatever pathway we're looking at or whatever process we're looking at is highly conserved between human and worm, and it might be well worth doing experiments. Like if TMED2, its role is unclear, we can do experiments in C. elegans, the corresponding ortholog here.